Hello, my name is Cana Quave and I'm a PhD student at the University of Texas Health Science Center at Houston. Today I'll be talking to you about neurons in the prelimbic cortex that encode the changing predictive value of reward-associated cues. So, in nature, animals must adapt to shifting of food availability in order to maintain efficiency during foraging. Loss of a previously available reward results in behavioral invigoration and ultimately strategy shifting. The mammalian prefrontal cortex plays a vital role in executive control of reward-related behavior, and in particular the rodent prelimbic cortex has been shown to play a role in processing detection of food reward cues, as well as in controlling behavioral flexibility and response shifting under changing conditions. However, it is unknown whether PL neuronal activity can encode fast changes in food availability. So, we hypothesize that PL neurons show modified reward cue responses that vary with changes in reward predictability. In order to test this hypothesis, we employed rats that had been trained to press a lever in response to audiovisual cues to obtain sucrose pellets, and we subjected them to 3 different behavioral protocols. In the first, we used semi-randomly intercalated rewarded and omitted cues in the unexpected reward omission protocol. And we also used repeated rewarded followed by repeated omitted cues, such that the omitted cues should become expected with time in the expected reward omission protocol. We also employed an extinction protocol with a long succession of omitted cues in order to induce behavioral reward cue extinction in these animals. So first, looking at behavior in the unexpected omission session, we saw that rats showed a frustration-like behavioral phenotype as evidenced by an increase in the frequency of lever pressing during omitted cues, as well as an increase in the maximum speed of locomotion in the period following the offset of omitted cues. However, in the expected omission session, we did not see this frustration, suggesting that animals quickly behaviorally adapt to reward omission once it becomes expected upon repetition. Now turning to our electrophysiological data, and in particular, in the unexpected omission session, when animals detected the presence of reward in the sucrose delivery dish. We see that cells responding with increased or decreased firing rates to reward detection did not respond to the detection of omission, and we see something very similar in the expected omission session, suggesting that PL neurons can encode either the presence or absence of reward through attenuated responses in firing rates. Continuing to look at detection responses, but this time responses to the repeated detection of reward omission. And comparing these same cells' responses to repeated reward, we see that cells responding with increased firing rates to omission did not previously respond to a reward. And when we look at the responses of these same cells, but now during unexpected omission, we see that they did not respond to unexpected omission either. And so a closer look at these data actually revealed that a select population of previously unresponsive PL neurons emerge to signal the detection of reward omission specifically when it becomes expected with repetition. Now, looking at cue responses during the repeated reward session, we see that while cells responding to reward cues with increased firing rates continue to respond to cues during expected omission, cells responding to reward cues with decreased firing rates show attenuated responding to cues once they fail to accurately predict reward during the expected omission session. And it should be noted that this effect occurred prior to the onset of behavioral reward cue extinction. So from these data, we predict that a loss of inhibitory cue responses in PL during expected reward omission precedes the transition to reward cue extinction. So now, looking at the extinction data, and in particular, cue responses, we see that both cells responding to reward cues with increased or decreased firing rates stop responding to cues during extinction. And if we look in the reverse, time-locking cells to cue responses during extinction, we see that while some excitatory cue responses persist in extinction, a small population of cells with inhibitory responses to cues emerge during reward cue extinction exclusively. From these data, we conclude that while cells responding to immediate reward omission progressively cease to respond when omission is repeated, a separate population of PL cells emerge to signal persistent reward loss. Secondly, real-time updating of reward cue responses in the PL occurs in conjunction with the changing predictive value of reward cues. Thank you for coming to my presentation, and I hope to see you at the virtual poster session. Take care.